this in Fiji, and it's called muscle motion. And this is a plugin that allows to measure the contraction of cardiomyocytes in different platforms. In this case, this is the paper about that they performed the algorithm, and they made experiments in order to standardize and get get the results in different ways. First, they mention how they perform the algorithm. The algorithm is based on image in bright field. The idea is measure the image or take the first image, and that the cardiomyocytes moves. They in the next image they recognize this move with the pixels that become dark because of the movement, and they use two different ways in order to get the algorithm calculate the reference image, and also you can put with manual in the algorithm. You can put, and they convert this. For example, the first is the amplitude, that is the size of pixels that move the cardiomyocytes, and this is the amplitude, and the other is the contraction, that is the this is the two things, that the amplitude and the velocity of these movements. And the algorithm at the end, the algorithm gives the amplitude of the cardiomyocytes and the velocity of the contractions. Okay, to standardize this method, as I told you, they use this first image and image reference, and the final the image results. They standardize with different type of cardiomyocytes. First, they use these isolate cardiomyocytes in monolayer, in organoids, and in patients, and they use the algorithm, and this is the contraction in this case, and in different cases, and this is the result of the algorithm that they get, and this is the amplitude of the contraction, and this is the velocity of the contraction. It means each peak represents different contractions, and the low peak represents the relaxation contraction. It means this is when it moves, when it's quiet, and when this move again. No? And this is in different type of cardiomyocytes. And the line red represents the error of this experiment. This is a linear regression about this experiment. Okay, and the second thing that they use is in order to standardize, or in order to see the experiments, use fluorescence in order to see if the algorithm adjusted with the predictions, or with the measurements. And then they use drugs in order to see what is the behavior of the cardiomyocytes, and they put these points. This is the value of the amplitude in the MSO, in the relaxation time that I told you, and they use different drugs. This is the name of the drugs, and they see how they decay this movement. No? And this is the way that you can see what is the behavior of the cardiomyocytes when you put drugs.
1 minute left.
Okay, and the end also, they contrast these results with another way the gold standard way to to measure these cardiomyocytes with electricity, and this is the results of in the up part. You see the electric the curve by electric performance, and this is the results. They use two different zebrafish. One is the wild type, that this is the normal behavior of the cardiomyocyte, and the second one is the mutation with defects in the cardiomyocyte, and they found by electric way different significance with the mutation zebrafish. Also in the algorithm, if you see, it's very close. And this is the velocity of the contraction normalized, and this is the velocity of contraction with electric way, and it's quite similar. And the final experiment, they use also two different. It's the same. The zebrafish. They use this contraction. This is the healthy, and this is the mutation that you see how they change the behavior of the cardiomyocytes. It means that this cardiomyocyte is disease, and it's the way that they use to standardize this procedure. Thank you. That is all.
Good morning, everyone. I will talking about this paper called NemaLife chip, a micropillar-based microfluidic culture device optimized for aging studies in crawling C. elegans. This study shows the microfluidic device that features an optimized micropillar leader in which worms can crawl, screening channels that separate the progeny and prevents the loss of adults during the maintenance of the culture, and ports that allow quick access to feed adult-only populations and introduce reagents as needed. To identify the optimal micropillar lattice device were designed with pillars that different diameters and edge-to-edge spacings, and the slow gate was measured in terms of wavelength, amplitude, and tracking speed. That in this device that was similar in the in the tree device. However, the tracking speed was higher in device 3, which has less strict confinement, and the workhouse in device have a locomotor gate that is more similar to that of serving plates in agar plates. For the optimizations of worms culture conditions, 200 microliters of S medium administered through the loading port of the device. All adults were retained and all progeny were removed using a total wash volume of 1 milliliter. To know the adequate food supply, they use a bacterial concentrations of 100 milligrams per milliliter of E. coli for once a day, twice a day, or every day, and to they observed that the worms feed every other day with 100 milligrams per milliliter had a general extension in the lifespan. To evaluate whether the worms could have a restricted diet, which is those of E. coli, the density of the bacterial solution was measured at the end of a feeding cycle. The residual bacterial density measured daily during the reproductive period, which decreased significantly after day 3 and stayed the same until the end of the period. Free microfluidic chambers. The half-life of worms grow in this environment was shorter and exhibited age-related valvar integrity disorders temperature and poor reproductive health. This suggests that the environment of the pillars can mitigate stress during the life of the C. elegans culture. They also compare the lifespan between the device on agar plates. The half-medium life were similar. They evaluated whether the chip induced environmental stress in the mutant strain that expresses the stress indicator DAF16. This strain was found to exhibit similar lifespan on agar plates and on the chip, indicating that the liquid environment of the device does not induce deleterious effects on survival. Um, Lifespan studies were performed using long-life genetic mutants, indicating that water stress due to daily washing feeding does not cause undesirable results. Finally, they also observe pharyngeal pumping, which is a physiological marker to assess the health status of C. elegans. Instead shows the pharynx of the worm in a chamber. The maximum pharyngeal pumping speeds on the chip were relatively slow in the microfluidic device compared to the agar. In conclusions, the NemaLife chip is a simple, low-cost means of obtaining reliable physiological and lifespan data in aging worms and has significant potential both as a handheld device or as an automated platform. That is all. Thank you.
Good morning. And today I would like to present this paper titled Parent Coaching Increases Conversational Turns and Advances Infant Language Development. Here, the researchers study 79 families, and they wanted their hypothesis was to assess if parent coaching intervention delivered at 6, 10, and 14 months would make a difference in the development of the baby's language. Okay, so first they divided these families into groups. The first group was the one that has this intervention. It means the parent coaching intervention. And the other group was the control group, which didn't have coaching condition. So the parental coaching intervention means that they are going to receive individual coaching appointments to receive linguistic feedback. And experts are going to suggest them, for example, to talk with the babies, like baby talking, and to while they are changing the diapers or when they go to groceries, and they are going to suggest appropriate age activities for the baby. Okay, so they assign these families of 6-month-old infants parent coaching intervention, And they also used these recorders, and they put the babies were wearing a vest, which inside the pocket it has recorder. So the parents are going to record their interactions with the babies at 6 months, 10 months, and also 14 months. Okay, so these are the results that they obtained. The parameters that they measure were the number of adult words over 12-hour day, also the conversational turn count. It means the response, the answer of the baby, and the vocalizations. Okay, so let's see how they related. Here you see that the percentage of parenthesis speech, the parenthesis speech means speaking to the baby in intonations. For example, the giraffe has long neck. So this tempo, this intonation, that's baby talking. So this parenthesis speech, you can compare here. The control group is the blue one, and the intervention parent coaching is the red one. So you can see clearly that when parents use parenthesis speech, here you see how it increased according to the age of the baby. Okay, and then we see here the conversational turns over 12-hour day. You can also see how it increased when parents has these intervention parent coaching. Then, if we see the child vocalizations over this 12-hour day, you can also see that it increased. The child vocalization increases according to the exposure to this interventional parent coaching according to the age. Okay. Also, they measure with these software and these algorithms called Lena. So the Lena recorders, then they use an algorithm to count the words that the baby vocalize. And here you see that the percentage of child words at 18 months, it's superior. Thank you, Andrea. This percentage is higher in the intervention group. It's higher than the percentage of child words in the control group. Also, they perform a survey to the parents and they see that these results are also higher for the intervention group than the control. Okay, thank you very much.
Hi everyone, I'm going to be presenting this paper. Submicrometer-Sized roughness suppresses bacterial adhesion. In this paper, they demonstrate that irregular 3D structure with submicrometer spacing can greatly suppress bacterial adhesion. In contrast to most strategies focusing on roughness, in this paper, the characteristic length scale falls just below the size of the bacterial cells. So, to investigate the influence of surface topography and surface wettability, 3 types of structured surfaces were incubated with bacterial suppression. The first one is a flat, perfluorinated glass that serves as a hydrophobic control surface. The second one, the D in the picture, is a superhydrophobic perfluorinated micropillar array that was used as reference surface that possesses microscale roughness. And the cylindrical micropillars were 5 micrometers in height, 13 micrometers in diameter, and the distance between the pillars was 20 micrometers. And the third is glass substrates coated with nanofilaments that show sub-microscale roughness. The surfaces coated with this silicon nanofilament enabled investigators to use this surface chemistry and surface wettability under otherwise identical conditions. The silicon nanofilament rendering were of approximately 50 nanometers in diameter, and they prepared 3 nanofilaments, super hydrophobic methyl-terminated nanofilaments, and then they activated with oxygen plasma, and these filaments became super hydrophilic. And then they also compared the addition of bacteria on fluorinated nanofilaments. So they used the surfaces that 1 centimeter by 1 centimeter, and they were fixed by double-sided tapes in a sterile chamber glass. Each surface was covered with a 5 micrometer thick layer of bacterial suspension that had E. coli expressing E-green fluorescence protein. And after 3 days of static incubation at 37 degrees, the suspension was replaced by PBS for observation under laser scanning confocal microscope under wet conditions. And after 72 hours, 3 days of stationary incubation, the perfluorinated glass, the flat one, was covered with several 3-dimensional microcolonies and showed a large number of bacterial colonies separated by regions almost free of bacteria. On the super hydrophobic SU8 micropillar array, the space between the pillars, the top faces, and the flat bottom were covered with individual bacteria and several microcolonies. And finally, in all surface coated with the nanofilament, only sparse and isolated bacteria was observed. The bacterial adhesion was consistently found to be reduced by approximately 2 orders of magnitude. So to gain insight into the long-term effect of these nanofilament-coated surfaces, the duration of the incubation was extended to 7 days. And even after more than doubling the incubation times, the surface coverage of the fluorinated nanofilament-coated surfaces remained below 0.7%. On the hydroxyl-terminated nanofilament, the bacterial coverage was even as low as 0.27%. And in contrast, the surface coverage obtained on the fluorinated glass, the flat glass, reached 10%, which is, again, 2 orders of magnitude larger. They also quantify bacterial adhesion using the plate count agar protocol, and the largest number of adhere colonies was found in the fluorinated pillars, followed by the flat glass. And again, the difference in the number of adhere colonies in all the nanofilament-based coating is 2 orders. Thank you. In the nanofilament-based coating is 2 orders of magnitude lower compared to the flat glass and the pillars. This also holds true for pillars with smaller diameter and spacing. Finally, to explore whether the nanofilaments are effective in delaying and preventing addition of other bacterial strains, they investigated spherical gram-positive Micrococcus lotus and rod-shaped gram-negative Pseudomonas fluorescens, and after 72 hours, all nanofilament coating surfaces with fluorine, methyl, and hydroxyl groups showed low bacterial coverage. So in conclusion, the design of nanofilament coatings with different surface wettability and modification supports the hypothesis of the importance of this 3D topography and the length scales of the coating. The spacing between the irregularly arranged nanofilaments falls just below the size of bacterial cells, and this has several advantages, such as the spacing is sufficiently small that the stiff bacteria cannot fit in between the filaments, and second, the spacing is sufficiently large to greatly reduce the number of possible addition points for the bacteria. Also, the effective anchoring area is further reduced by the local curvature of the filaments. So this is my paper. Thank you.
Okay. My topic is Wort substrate consumption and metabolite production during lambic beer fermentation, and maturation explained the successive growth of specific bacterial and yeast species. The present study combined the sending in higher cultures and a metabolic analysis to decide the identify evolution and function of microorganisms, substrates, and metabolites during the fermentation process and maturation of 4 phases of lambic beer production. The fermented wort and the maturing lambic beer cross several transitions between microbial species. Manual acidification of the moss with lactic acid shortened the enterobacterial phase. And the species consumed acetoin produced by acetic acid bacteria for redox balance. In this part, two lambic beer production process were studied as a function of time, pH, temperature, and attenuation of the moss before its transfer to the barrels was 4.3, 17.1 and 12.6 respectively, and the pH decreased during the first 2 weeks. In this part, cyclohexamide resistant yeast were counted, work with acetic acid bacteria, lactic acid bacteria, yeast peptonagar, mannitol-agar, and yeast peptonagar plus cyclohexamide. In this part, during the 24-month lambic beer production process, several bacterial species were identified: Klebsiella, Enterobacter, Lactobacillus, Saccharomyces cerevisiae, Picha, and this part, the number of yeast sensitive to cyclohexamide reach a maximum after 3 days of fermentation and remaining stable for 3 weeks. Through the samples taken in the fermentation, most male species of Saccharomyces and cellular C. microbium. During the first month of fermentation, the appearance of cell overlapped with the enterobacterial phase, and last 2 more weeks. During the last weeks of the main fermentation phase, the most predominant yeast Saccharomyces cerevisiae. And this part, the initial most was rich in carbohydrates, including fructose, 2.5 grains per liter, glucose, saccharose, maltose, maltodextrose, and
1 minute left.
oligosaccharides malt. Organic acids were also present, among which were lactic acid, citric acid, malic acid, and gluconic acid. And the conclusion, the present study monitored the temporal and spatial distribution of microorganisms, substrates, and metabolites during the production process of lambic beer, and is a new understanding of how microbial communities are configured throughout the production process. Thanks.
Good morning, everyone. Excuse me.
Ramiro.
I have a problem with my share.
Okay, we can continue with the next one until Ramiro solve the problem.
Next. You can see my screen.
Yes, we can see it.
Okay. Hello, everyone. In this occasion, I try to explain this paper, but proving product growth, metabolism, and reciprocal toxicity when integrated human into in organ on a chip platform. Okay. This paper is very interesting for construct this setup where use different surface treatment of in this case use a 2 glass for compact the channels or chambers, okay, and put inside different cells and organs, okay. You can see in this and the continued cell culture in the platform on a chip for the approved drugs, okay. And they try to prove different drugs in a primary human culture, and in this case, for example, cardiac use in using human pluripotent stem cells and put in the macro device on the system, okay, and after compare the ability and different treatment for a long time on different conservation. For example, you can see the organ on liver and cardiac, different lung, endothelium and brain, but use the principal liver cardiac organ. Okay. After they grow this organ in a micro device system and prove that different drugs, and you can see in control drugs and after drugs in this image on fluorescent microscopy, and you can see how drugs make the metabolism and is induced of apoptosis or different signal for necrosis or other signal in organ. Okay, you know in this type of organ the drugs is lower in a normal or conventional cell culture in 2D. For example, when I difference cardiac cells in a normal or conventional cell culture, but in this case on 3D dimension and reaction is different. They make viability and prove different tissues and organ, but prove different treatment and the concentration in the conventional, for example, use in the conventional hand drop, and compare with microfluidic treatment and use this concentration and make a different strategy for calculating the viability cells, right? And you can see dislocation and a liver and low in the first condition, and after use induce apoptosis. Okay, and after they implement the high viability for drugs. Before I talk about this, the different reaction of conventional grow on the different in 3D dimension for organ. Okay, this next experiment and show the figure and the toxicity and the different concentration, and after other tissues, for example, brain, and reaction and different of control or different.
1 minute left.
Okay, thanks. And the different condition for expose the organ. You can see how the organ is disintegrate in a lab on time. Okay, this other conversation, how different tissues. You can see that the three principal or the other testing. This investigation concludes in this study. It's very easy demonstrate the toxicity for drugs in continuous cell culture, macro device, and 3D dimension organ for studies. It's a powerful tool for investigate primary drugs and in primary tissues for human. That is all. Thanks.
Okay, I'm so sorry for the problem. Now I'm going to present this paper called Laser-Induced Fluorescence with 2D Hilbert transform edge detection algorithm and 3D fluorescence image with white spot early recognition. In this paper, the authors abort the with the objective of recognition dental problems in people. So in this case, the algorithms are focused in image processing acquired by fluorescence. In this case, the laser used is the low energy in range of 5 milliwatts. The principal idea of this study is abort the reflection of wavelength in order to represent a modeling to detection of caries. So in this image, the author presents the weight of the light source influence in the tissue. In this case, for example, the absorption and transmission is detected for the diffuse reflectance. So according to different characteristics of surface, the wave behavior for this wave, for example, in this case, the different tissues are represented for the reflection of laser signal. In this image, for example, white spot, which is the objective of this research, is described with the highest sound of peak. In this image, for example, the wavelength are 633 nanometers. So with this characteristic, the white spot early detection is focused to avoid the early detection of caries. Reflection of laser wave is represented according to this image, which represents the wavelength of detection. For example, in this image, the caries are detected in the red color, and the white spot in the same way. Next objective of this research is avoid the edge detection. So in this case, the signal are compared with your wavelength. So in this case, for example, the image representing is according to these images, which is describe the reflection of the laser signal. So in this image, for example, white spots are represented with a big area, and the caries or stain are represented with a low area from this high spectral images. This behavior are represented with a 3D images, which is the 3 dimension and represent the reflected signal from the surface. So finally, the principal objective is detect the white spot in order to prevent the future problems in dental. The principal purposes of this research is use the Hilbert recognition for describe the behavior of the dental problems. That is all. Thank you.
Juan, are you there? You are the next one.
Yes, but I have a little problems, but I guess I can talk by the phone.
Okay. Can you reach a computer while other start the next presentation?
Yes.
Okay. Who is next? Ramiro, could you solve your problem? Oh no, yes, your spot. Who is in? Eric got the shoot yesterday and told me that he will not present. Luis had bad connection. I don't know, Luis. Can you present?
Good morning. In this moment, I don't have internet connectivity. I am using my phone here.
Okay.
I prefer to explain the next week. My paper studies the spontaneous booking of action using poroelastic sheet, and I wanted to show us that from a physical model.
Okay. Next week you can present. Who is next? Edison. Oh. Okay, it's just Juan left.
Yes.
Yes.
Okay, can I present this paper, my paper?
Yeah.
Okay. Sorry about this problem, but
no problem.
Okay. Yes. Everyone can see the screen, my screen?
Yes.
Yes.
Thank you. So, okay, I'm going to present this review. It's about microfluidics for cell factory and bioprocess development. Well, the microfluidic tools are being employed in cell factory and bioprocess development to speed up development and address new products. For example, microfluidics for bioprocess development range from integrated device for DNA assembly and transformation to high throughput screening of cell factory libraries and micron scale bioreactors for process optimization. So the microfluidic tools take advantage of control at the micron scale and the scale of the single cell, and utilize the unique fluid flow characteristics at this scale to improve throughput in some handling and facilitate automation and enable new assay modalities. This process follows 4 steps: engineering, design, building, test, and evaluate the cycle of this process. The first step is carried out in silico and focuses on the design of libraries of cell factory variants. The microfluidic components and technologies can be tough and of us approaching cell factory and bioprocess development either from the bottom up or the top down perspective. So the bottom up means that starts from the single cell methodologies such as picoliter scale and applies these concepts to bioprocess related questions. And the top down is the approach, on the other hand, and starts from the full scale reactor and attempts to miniaturize its size while preserving its functionality. You can see a schematic process of this development with and without the use of microfluidics. So by introducing the microfluidic technologies, more strains can be constructed, sorted, and evaluated while spending less time and resources the right of the figure, you can see that. Okay. The evaluation step in the bioprocess development consists of characterizing selected candidate strains from previous steps and optimizing cell factory function for scale up to production scale. Employing microfluidic technologies that allowed more strains to be thoroughly characterized in parallel. Substantial efforts have been put into the top-down approach of developing miniaturized bioreactors focusing on the range response of colonies or cultures rather than single cells, and is of great importance for culture process. The cell factories are ideally cultivated using fed batch, chemostat, and perfusion bioreactors. In order to miniaturize these reactors, the cells have to be maintained in one area while media is supplemented and removed. So in recent studies, an even more complex microbioreactor developed is a multi-phase microreactor for continuous chemostat cultivation of yeast using microbubbles for aeration and liquid convection.
1 minute left.
And finally, these microfluidic bioreactors mimic the culture conditions in full scale and allows for the collection of rich bioprocess data with a reduced media consumption and increased potential for parallelization. Thank you.